From my work in Germany, I continue doing work on the microbiology and enzymology of phytases and carbohydrates, and with the focus on application on feed industry. So, with my colleagues in the marine sciences, namely here, Dr. Sergei Dobretsov, Professor Sergei Dobretsov, he was working with these aquaponic systems, and in our cooperation, we found that it was very interesting to study which microbes are being responsible for the nutrient cycle in these systems. And especially we're interested in microbes carrying phytases and enzymes that degrade fiber, non-starch polysaccharides. This work was performed in the lab by Nihad Al-Marouki. So what is aquaponic system? Why are we interested in that? Aquaponic is an association of hydroponics and aquaculture. Yeah, and the hydroponic system is a solution-based growing plants, a soil-less way of growing plants. It's aquatic-based environment where you use nutrient-rich solutions applied directly to the roots, and you have to provide some substrate for the support of the roots of the plant. So we are interested in nutrient cycling for the plants in this system, and where these nutrients are coming from. These nutrients are coming from the aquaculture side of the aquaponics. Yeah, the aquaponics is linking a fish tank and an aquaponic system, and the fish is unable to digest all the feed and absorb all the nutrients from the feed, and therefore it's secreting a lot of nutrients into the water. This is a highly contaminant activity, and the association of this system with a hydroponic system allows for using those nutrients and having a double purpose activity where you can generate profit both by the crops and the aquaculture systems producing the fish. So we were interested in analyzing the systems for the microbes that are carrying out the nutrient cycling, specifically for fiber degradation and for phytate degradation. So phytate is the molecule, my pet molecule, the things that I studied all my years since PhD up to now. And phytate is the main way in which plants store phosphorus in seeds. Seeds are used for making feed for animals, including fish. So fish feed contains a high amount of phytate. Nevertheless, the fish lacks the enzymes necessary to degrade the phytate, and the phytate is excreted into the water, and therefore must be, if the plants are going to uptake this phosphorus bound to the phytate, this phytate needs to be hydrolyzed by phytase enzymes, and actually there's a huge amount of phosphorus that comes out of the feed, undigested feed or uneaten feed on these systems, and it's only polluting the water. In the aquaponic systems, the microbes, of course, they will adapt to the availability of this nutrient and start degrading it and hydrolyzing it into phosphate, and the phosphate will be able to be uptaken by the plants. Similarly, the fibers are a carbon source for microbes. They're highly unavailable for the fish, but the microbes will likely use it as a source of carbon and energy. They're heterotrophic microbes in this aqueous aquatic system, and they are using the carbon from the fiber as a source of energy and carbon. But in that process also, they are able to mineralize the organic matter and release other nutrients bound to this organic matter, such as nitrogen and sulfur. So the objectives of this study were to investigate the diversity of bacterial communities on these systems and identify which part of this community is responsible for phytate mineralization and non-starch polysaccharide mineralization. We used barcoding, 16S barcoding analysis for mapping bacterial communities, both in fish compartments and in hydroponic compartments. And we also did screening for bacteria and carrying these traits. And we characterized their activities for hydrolyzing both phytates, cellulases, xylanases, glucanases, in beta-glucan, xylan, cellulose, and phytate. So here's what the sort of the systems looks like. These are demonstrative systems used to teach the students on our department. And we have associated side by side where you have fish compartments and plant compartments. In total here we have 8 systems. We chose 3 systems that were in similar conditions with quite a lot of biofilm grown. And we use these 3 different systems as replicates for these studies, for this study. Here's another view of the fish compartment on the left. There is a filter in the middle, which is just a biofilm system, and the plant hydroponic compartment on the right. So the samples were collected biofilm in water, and they were homogenized. DNA was extracted using PureLink microbiome kits from Fisher Scientific. The DNA extracts were sent for sequencing abroad because at this moment we didn't have the equipment for DNA sequencing in our lab. Now we have nanopore MinION system. But at that time we didn't have any way of performing this sequencing, and we used the service from Mr. DNA in Texas, molecular biology lab in Texas, USA, for the V4 sequencing and pipeline for taxonomic assignation of zOTUs from the sequences. We compare those OTUs that are found by the 16S metabarcoding with the databases available in the FAM webpage. These databases are for species. They are reported to carry homologs for the genes of these enzymes that we are interested in. So they are a soft evidence for the presence of this trait. So the more OTUs that we identified that were previously reported to carry the genes for these enzymes, this is a reflection that there is a higher likelihood for this trait being present on this environment. So we compare our metabarcoding with this, with the databases available for the distribution of these enzymes across the tree of life. So we also did work by isolating bacteria from the system and screening for carrying these enzymes. And the isolation was done after enriching samples with wheat bran, because wheat bran will carry both phytate and fiber. And this was incubated for 3 days before isolating by standard culture methods. And isolated strains, 39 isolated strains were stored in glycerol for subsequent assays. So we did the identification of only for the strains that were identified positive for some of the enzymes here. And for our surprise, most of the microbes here were bacilli. All of them were bacilli actually, but most were from Bacillus genus, we still have one Staphylococcus and one Rosemollera acumaris, but two Rosemollera. Anyway, but actually the majority of the strains were bacillus, which indicates that these the isolation method that we used was highly selected for this type of microbes, or that is these microbes are responsible for the cycling of these this type of compounds on this system. So this is having a quick look on the screening of these strains for their ability to hydrolyze starch and fiber. Starch is stained by grams iodine, and fiber assays are stained by 0.1% Congo red solutions, and they form these halo structures, which indicates that they have hydrolyzed the fiber in the agar plates. For the phytate, it's a little bit more complicated because the agar plates, they tend to provide a huge amount of false positives. So we have to move to detecting this strains by assaying the broths for the extracellular inorganic phosphorus concentration. So the disadvantage of doing this is that you are not detecting the enzymes that are intracellular. But in our case, since this study is a study about cycling, nutrient cycling, the intracellular phytases are not really going to be responsible for nutrient cycling in this environment. So we are in the correct approach to ignore them for the moment. We tested the changes in extracellular phosphorus concentration, and the strains that have negative balance, it means they're absorbing the phosphorus from the broth, whereas the ones that have positive balance are the ones that are releasing more into the solution than they are absorbing. So the ones that cross the threshold of the limit of detection, we consider them a positive and assay them with the kinetic, just to make sure that we have a real positive. So we have 8 positives here, and then after reassaying, we have only 2 positives. So we really made sure that we had the correct strains screened for this trait. So this is how it looked like the snapshot of the microbial community. On the left, we have the fish compartments, and the right, you have the plant compartments. There are huge changes on the microbial community. Nevertheless, so surprisingly, the diversity index are very similar. So we have that for the fish compartment, we have that you have 527 OTUs that are exclusively uniquely occurring on that plant compartment. That corresponds to 27% of the total OTUs found, which is a very high amount. And in the fish compartment, 296 OTUs were exclusively found on the fish compartment. So the community structure is significantly different, but because of high variability of the community found between the replicates were not very similar to each other, then it became really hard to prove this statistically, those differences. So we did some clustering with a very cool app called Graphia, and this clustering of the OTUs in these systems was performed based on correlation. So this analysis called the network correlation analysis, and the samples that are linked together, each OTU or group of OTUs that are co-occurring, they are represented by one small nodule here in this system, and the closer they are together, the higher the correlations that when one OTU is increasing, the other is also increasing. And the further away, they're more unrelated to each other. So by doing this analysis, we found that the clusters 1, 2, and 5 were representing the fish compartments, and clusters 3, 4, and 6 were representing the unique microbes that were occurring on the plant compartment. Okay, thank you. So we also checked for the web page for the presence of these OTUs that were previously reported to carry these enzymes. And we found that there's a lot more of these of carbohydrates reported for these microbes than phytases. Among phytases, beta-propeller phytases were the catalytic mechanism more abundant, which is coincident with our findings from or isolates. And among the carbohydrates, the cellulases were the most common ones. Among our isolates, we found that only 2 phytases were strains were positive for phytases after confirmation, and among the carbohydrates, the cellulase-degrading enzymes were the more ubiquitous, followed by amylases, beta-glucanases, and xylanases. This is the confirmation of the kinetic of phosphorus release by the 2 positive strains for phytase, not bacillus. They were staphylococcus and roseolum, and this was surprising for us because we were these enzymes on this system. So to conclude, we had a very diverse community on these systems. We could not find statistical difference between fish and plant compartments due to strong variations in among the replicates. The cellulases were the most common enzymes on these systems, and phytases were rarely found, and they were found to be beta-propeller enzymes on the systems, which is a little bit disappointing because beta-propellers are not great for biotechnological applications, which is what we are intending to study these enzymes for afterwards. So still there is a huge potential for isolating the other microbes that we have not found on the systems carrying histidine acid phosphatases and trying to explore them for biotechnological applications and feed additives later on. So I just want to acknowledge the grants that finance this work, and thank you all for your attention.